one of the grand challenges of science is to discover and document the biodiversity on the planet and understand mechanisms that underlie that diversity. And in recent years, advances in sequencing technology have really helped us jump forward with our understanding of biodiversity. The workhorse platforms have given us a good understanding of regions of the genome that you can readily assemble from short-read data or that are compatible with PCR methods. Those approaches don't let us look at the highly repetitive regions of the genome very well that are abundant in many organisms. And as long-read sequencing gets established, we're starting to learn, especially for model organisms, that these highly repetitive regions are really critical to regulation, maintenance, and genome evolution on many different scales. And so I'm excited about trying to take signal from these repetitive regions and see what they can teach us in diverse non-model groups. I got excited about repeats near the end of my PhD. I was studying a species group of ground beetles called the Brevi species group. There were 2 valid species in the literature for a long time. We had evidence that cryptic species were present, and so for a lot of my PhD, I was taking a pretty traditional species delimitation approach where I gather material from the field and museums and generate molecular, geographic, and morphological data sets, and then use multiple lines of evidence to formulate species boundaries. The result of that work is that I found good evidence that at least 9 species were present in the group. And right at the end of that project, one species in particular presented a challenge that got me excited about repeats. And there was a type specimen, Bembidium levigilum, and we knew that that name should apply to one of our species concepts, but we couldn't confidently identify it because it was a female and it was 100 years old, and the best characters are in the male genitalia for this species pair. So we knew if we get sequence data, maybe we could place it to species. And so I designed a study to get sequences out of this and other 100, 150-year-old type specimens. Most of the other type specimens worked great, but the data from this was the one specimen that gave us really degraded sequences. And with our target genes, we couldn't confidently place it. We had a little hint, but weren't sure. And so what I did is I got a reference sequence of nuclear ribosomal DNA, 14,000 bases containing 18S, 28S. And I mapped all of my Illumina reads to that reference sequence. And I saw a surprising result when I looked at the coverage depth, or what I'm going to refer to as a copy number profile, of the resulting read mapping. And so what you can see is that for the first half of my reference sequence, I had really miserable coverage. But then partway through, from one base to the next, I suddenly had 400x coverage from this 100-year-old specimen. And my initial reaction is something bad happened. There was a fungal contaminant in the sample, or I've got an assembly issue. And I ruled those things out, and I went and got a fresh specimen of species A and species B, and got low coverage Illumina data, and I made profiles. And species A had a profile in which, in the exact same region, there was dramatically inflated coverage as the type specimen, and that was cool. Species B had a very different looking profile. And I'll point out that the y-axes are scaled differently on these 2 profiles. So even though this looks like it's got lots of copies, this is equivalent to this little skinny gray line in species A. And it's just that in species A, there's just really dramatic inflation of these sequences here. And so in the short term, I was excited because it gave me a lot more confidence in assigning the name of that type specimen. But what was really exciting to me is that of all the Illumina reads that I got from this fresh specimen of species A, 17% of all genomic reads were mapping to this one reference sequence. In the other species, it was less than 1% and consistent with what you might expect for just total ribosomal DNA content. And so that was exciting because I just spent 3.5 years trying really hard to see signal in multi-gene data sets that would help me separate these 2 species. But these data suggested that there's likely really rapid and major rearrangements or evolution happening among the repetitive sequences in these 2 species. I went and sequenced the 3rd species. It also had a highly inflated region, but it had kind of its own signature. And so that kind of sent me off on a study in which I wanted to ask the question, are there species-specific signatures in the species of this group when I look at these profiles? And the answer is there are species-specific signatures. So here are the 9 species in the group. And I was showing you gray copy number profiles, and now I'm going to show you profiles that have a color ramp applied, which indicates a standard scale of copy numbers. So if you see a blue profile or a profile with blue regions, there are fewer than 2,500 copies. If there's any red in the profiles, they've got 20,000, and in some cases hundreds of thousands of copies. So what was cool is that there are species-specific patterns, and there are several species that have, for some reason, dramatic inflation. When I first saw this, I had no sense for why they would have such different profiles and what could be causing these regions of inflation. And so I went to the literature, and it turns out that cytogenetic folks for years have been mapping the location of ribosomal DNA on chromosomes. And in plants and animals and fungi, there are dozens of papers saying, hey, sometimes a little fragment of ribosomal DNA can escape the functional ribosomal clusters, land somewhere else in the genome, like in a heterochromatic, highly repetitive region, and they just get massively duplicated. And so it seems that we're seeing with sequence data, we're able to visualize that phenomenon with short read data and low coverage, which was kind of a cool connection to make. The next question was, if we take multiple individuals per species, are these signatures that we see stable? And so I'm showing you now, again, the species tree that I had generated as part of that delimitation project. And I'm using that as a framework to ask about the stability of these profiles. And I've shrunk down the profiles small, but hopefully that you can see, for example, in this green species, every profile that we sampled has the signature of an inflated region over in 28S, and that signature is very stable. And as we go species by species, we see repeated patterns of really stable signatures that are present across multiple individuals from diverse geographic localities. So you can see that just by eyeballing it. If we look at it statistically, this plot shows pairwise comparison of profile shape among all of our samples. And we've got within species comparison of profiles colored in orange, and between species comparisons are in blue. And so we've got correlation values, row values near 1 for all of our within species comparison of profiles. And between species show a wide range of lower correlation values. If we zoom in on this one clade and look at all of its profiles, there's a couple other insights we can gain. So this clade has some geographic structure. The specimens that are shown by circles are western distributed, and that clade is distinctive in the phylogenetically from this eastern clade shown by stars. And if we just ask the question, in this inflated region, what's the maximum number of copies present? In general, there are fewer copies in this western clade than the eastern clade. The least copies we observed were 47,000, and this specimen in the eastern clade had 4-fold more total copies in that inflated region. That within population and within species pattern of fluctuation of absolute number of repeats, that's something we expect based on repeat biology. And that's because when you have tandem arrays of repeats, recombination can cause unequal exchange, and in one recombination event, these clusters can shrink and expand really rapidly. So we know repeats are really dynamic at the species level. What's kind of cool, though, below the species level, what's cool is that we get a signal when we visualize the data in this way that's very stable, and that's that the boundaries of regions that are inflated, they're just invariant across every species. And again, that's because we think in the evolutionary history of this group, a fragment of ribosomal DNA mobilized, it gave birth to a new repeat unit elsewhere, and that became a fixed signature in the species. And we think that it happens rare enough that it does fix within species in this group, but it's frequent enough that when we sample lots of species, we see it happening independently, repeatedly. So we wanted to validate the sequence-based patterns, and I designed a fluorescence in situ hybridization, or a fish experiment. And if I make fluorescent probes in an uninflated 18S region and hybridize those to the 24 chromosomes, it lights up 2 chromosomes. And that's presumably the location of functional ribosomal clusters. If I make a fluorescent probe in the inflated 28S region and hybridize those probes, we've got ribosomal-like sequences lighting up every single chromosome. And so that validates the notion that between these closely related species, the profiles are telling us information about rapid genome evolution in the repetitive component of the genomes. When I analyze the regions, the sequences in these inflated regions with predictive software that tells you what kind of repeats you have in your samples, they're always annotated as satellite DNA. And that makes a lot of sense. And if we think about a greater context of satellite DNA, for a long time we've known that many chromosomes and many organisms have these tandem blocks of satellite DNA out in the telomeres and or in the centromeres. And if you've got a chromosome with a specific flavor of satellites and it's evolving along in a lineage, and there's an event that separates a subset of those organisms in terms of gene flow stopping, it's been shown that in a lot of work in model organisms that you can have rapid turnover of the satellite DNA composition. And in Drosophila, for example, over a 240,000 year time span, clades have shown rapid turnover in a species-specific way. And what you've now got, if that happens in this lineage, is a homologous chromosome that has blocks of now non-homologous sequences that are in, sometimes important regions of the chromosomes. And so in terms of a mechanism of reproductive isolation, if these come back together, there's potential mechanisms that are related to speciation here. So in conclusion, I told you that in this species, these repetitive DNA profiles, they really tightly reflect the species boundaries that we were able to come up with from a separate study. And they might be a really great source of data that can be informative at the species level. And not only that, we've been able to show that they show evidence, they can show evidence that there's been rapid genome evolution in the repetitive components of the genome that you just don't see if you look at large data sets of genes that require orthologous, assumptions of orthology and just the traditional methods. We don't see this pattern. And finally, it may be that the patterns that we see in these profiles are related fundamentally to mechanisms that are tied in with reproductive isolation and the process of speciation. I've got a pipeline underway with the help of a great undergrad named Sharif. We're trying to automate this process for any repetitive sequence and low coverage Illumina data.